And now for something completely different. Today I'm going to talk about data presentation, how to make a good plot, and how to report numbers in publications. This is an example of a good, decent plot. There are all the elements of a good plot are here. There are axes, there are descriptions of axes, there is a unit here, hours. There are data points, arrow bars, there is some model fitted to the data, and finally there is a caption that explains everything, including what sort of arrow bars these are. They are 95% confidence intervals. It explains what sort of function was fitted to data. There are three general rules of making good plots. The first one is clarity of presentation. You want to present a clear figure. You want to deliver certain information in clearest way possible. And this information has to be complete, but not redundant or obfuscated. The second rule of making good plots is clarity of presentation. And you can guess the third rule of making good plots is clarity of presentation. Remember this. This is what you need to do every single time. First of all, you need to show your data. This example is bad. It's actually awful. This is something which I call dynamite plot. But this data comes from an experiment where we have two groups of mice. We measure their body weight, and these groups are wild type and some sort of a knockout. Unfortunately, this type of plot, as you can see it in literature quite often, and it hides a lot of things. It hides data, it hides data variability. Don't do it. This is slightly better. What you see here is a box and whisker plot with some outliers. You can see the median, you can see interquartile range. It's okay. Perhaps even better, it's to show all your data because, if you have only a couple of dozen data points, it's very easy to show them in one plot. The problem with this plot, which is called a jitter plot, where data are randomly jittered in x-axis because it's a categorical axis. The problem with this plot is that some of the data points overlap and it's sometimes hard to see where they are. There are better ways of doing this. This is called a B-swarm plot. It's essentially the same type of plot, but there is a clever algorithm behind it to scatter data in a way that the data points do not overlap in the plot. And this plot shows all your data. It shows distribution of data. There are more data points here, fewer up and there. So the first thing when you look at this, you notice is the distribution of data, how they are distributed, and you can compare the two sets immediately. This is not difficult to do in R. I will leave you with this. You can come back to your handouts and check this. This is the code that creates all the four panels from the previous figure. The main ingredient of most of plots, types of plots, are lines and symbols. Have a look at this panel, the first one. These symbols are completely unnecessary. They look like asterisks and they are messy, they are destructive. It's much better to plot it as very simple symbols, just dots or little circles. So simplicity helps a lot. In the second case, you have simple symbols, just circles, but there are two data sets, and it's very hard to distinguish which points belong to which data set. It's much better done when you mark them with different colors. The third plot is probably quite obvious. I used very large symbol size. There are big squares overlapping, and it's very hard to see what's going on. In the bottom panel is exactly the same data, but plotted using much smaller symbols. The next one is a bit tricky. You have two data sets that overlap in this area and they become different. So this plot is probably okay-ish, but we can make it better. Instead of points, I used a line connecting them. It looks a little bit cleaner. You can see how they follow the trend. And also I changed colors. Red and green are not the best colors for colorblind people. There are better color palettes that can be used, and these two colors are easier to distinguish for the majority of population. Finally, this is really bad. There are several data points in two sets. They are plotted with different colors, which is fine. But the problem is they are joined by a line, the so-called spline. This is a way of interpolating between data points. The problem is that this suggests that there is a model that fits between data points, especially that the model peaks here. There is the highest value, not this point, but this one. It's much better if you have to join your points just to guide the eye. It's much better to join them with lines like this, which do end near the point and leave a little gap around each point. This looks better, and this does not suggest any values in between points. The next suggestion I'm going to make is to make your plots colorblind friendly. Color blindness affects about 8% men and about 0.5% of women. And depending on the color blindness type, people have difficulties distinguishing some of the colors, not all of them, but some of them, quite often red and green. So using red and green in the same plot is not the best idea. This is an example. These two panels, left and right, show exactly the same data. There are some mice data where body mass depends on the daily calorific input, and there are three strains, S1, S2, and S3. In the left plot is the default R color palette. If you use ggplot in R and make this plot very simple, don't change defaults, this is what you are going to obtain. There are three colors, red, green, and blue. And unfortunately, there are people for whom red and green are rather difficult to distinguish. So all these points will look pretty much the same. That's why instead of this, I use quite often what is called an Okabe-Ito palette. You can Google it out, you can read about it. It uses different sets of colors. It's down there. If you want to use it in R, you can copy this from here. I don't have time to go into details how to do this in R. You can find it. Again, Google is your friend. It's not hard to do, but this plot is much better. People with color blindness can distinguish between these three colors better than in the plot on the left. The next thing is about labels. This is a type of a plot you can occasionally see in a seminar, talk, or a lecture, where the person presenting this talk was too lazy to create a new plot. They just copied it from a paper, lost some resolution, and eventually you get a plot where you cannot read labels at all. If you have a plot like this and you don't have a better version, I'm afraid the only thing to do is just to replace it with your own better version. Just replace the axis, put some effort into it so the viewers of your presentation can actually read axes. Make sure they are visible. Next thing is logarithmic plots. I talked about log normal distribution before, and I mentioned that it's much better to present log normally distributed data as a logarithm or using a logarithmic scale. You can see the same data from sample 1 and sample 2 comparison here in linear scale, and this is one of the samples. This is the distribution of these data, and these two plots hide a lot. First of all, most of the data are here, so most of data points would be somewhere here. You can't really see them. You only see data points with highest intensity. If you take a logarithm of data, it looks much better. You can see all data points, and you can see the distribution of these data resembles Gaussian or normal distribution. There is something going on here at lower counts, but nobody is perfect and no distribution is perfect. Another example, you have a time course experiment when you measure some sort of abundance over time, and this is raw data with error bars. If you plot it in the logarithmic scale, this is log 10 of abundance, you will immediately notice two things. First of all, in logarithmic scale, these data follow almost a straight line, roughly. You can fit it with a straight line quite well. That tells you that your growth here is exponential. The second thing you notice is the arrow bars here are larger at the end and much smaller here. So arrow bar changes a lot. However, here all arrow bars are very similar. And this is typical in many types of experiments where the size of an error bar is proportional to the value of your data. And in logarithmic scale, it looks better. Error bars are comparable, data follow very nice relation. On the right, you have two panels that compare two sets of data, a wild type and a knockout, and there is some cell count here. And if you make a bee swarm here, this one is very flattened because the algorithm tries to scatter points around so they do not overlap. This plot shows what you need to see. Indeed, knockout is much lower than the wild type and is probably fine. But I would argue that it's better to show this in logarithmic scale. Instead of using a logarithm of data, I'm applying now a proper logarithmic scale. You can do this in R. There are tools to do this. So you see that scale is not even. There is 10, there is 100, 1,000 would be somewhere here. So the equal distance correspond to 1 decade in data from 1, 10, 100, 1,000. And in logarithmic scale, data look more evenly distributed, more normally distributed, I would say. How do you plot error bars? Well, it seems straightforward, but let's have a look at some details. This is our value axis. We measure something. The measurement, the actual measurement is represented by this point. There is a plus and minus error bar, and that could be a standard deviation, standard error, could be a reading error. There are many possibilities. There is a cap just to visually help finding where the end of the error bar is. If you want to present a confidence interval, then you make exactly the same plot, but the confidence interval extends from the lower value to the upper value, like this, from ML to MU. Confidence intervals, as you know, can be asymmetric. If you find a confidence interval of a proportion or of a correlation coefficient, they will be asymmetric. And in this particular case, upper error is smaller than the lower error. And you can plot it. You can still plot it like this. Errors do not have to be symmetric. And if you want to write it down, you have to write it as M plus R upper error and minus lower error. And there will be two different numbers here. Again, you have to think about clarity when you make your plots with error bars. Sometimes you can plot just normal error bars like this with a little cap at the end. Occasionally, you might have error bar in both axes, X and Y. You can plot like this. When you have lots of data points on top of each other and the plot looks very dense, you can simplify things. Again, simplicity is the key. And in this particular plot, if you look carefully at error bars, they are not like here, black and with caps, because that will be very messy. I removed caps, the error bars are now just lines, and I also made them gray. They are still visible, but they don't distract you from the main data. And finally, if you need to make a bar plot with error bars, make sure both arms of the error bar are visible. What sort of errors can you plot? You already learned about standard deviation, standard error, and confidence interval. And it's just a quick reminder that standard deviation shows the scatter in the sample. And you can use it when you want to compare two or more samples, though probably box plots or even B-swarm plots are a better alternative. Instead, if you really want to show scatter of your data, the mean with plus minus standard deviation makes sense in complicated plots when you have lots of data, and showing data points, original data points, would be very messy. Standard error is the error of a mean, and it's most commonly used error bar. Though confidence intervals have better statistical intuition, so if you want to use a standard error, I suggest try to calculate confidence interval, like the 95% confidence interval, and use it instead. I would argue that this is the best representation of uncertainty, and it can be used in almost any case. And remember, always state what type of uncertainty is presented by arrow bars in your plot. It's very important. Box plots, I'm guessing you are familiar with them, but just a quick reminder. Box plot is a way of representing multiple data points in one simple visual object. You take a data set, hundreds of data points. They are on this axis. There will be median in the middle, which is the 50th percentile. The box will extend from the 25th percentile to the 75th percentile. That means 25% of data are below this point and 25% of data are above this point. 50% of data are within this box. So the distance between the ends of this box is called interquartile range. This is yet another measure of data dispersion, which you can use instead of standard deviation. The box is quite standard, and most of box and whisker plots will have exactly the same. However, the whiskers are, well, they depend. Sometimes they are 95 percentiles. Sometimes, like in R, they extend up to the 1.5 times interquartile range. Occasionally, it might extend beyond the last point in data. That's why it says less or equal 1.5% of IQR, because if the whisker extends farther out than the last point, it will be trimmed to the last point. If there are points outside whiskers, they are plotted directly as the so-called outliers. Box plots are a good alternative to standard deviation arrow bars. They are a non-parametric and show pure data and useful for large number of data points. On the left, you can see an example of a box plot with 3 boxes, and the fourth one, just as a comparison, I created as a bee swarm plot with the median inside. This is a good alternative for box plots, and in many cases, it's better to show all of your data than reduce them to box plots. On the other hand, when you have a plot like this is a sequence quality from a sequencing experiment. This is a position in the sequencing read, and this is a quality measure. And each box contains millions of data points. So you can't use a B-swarm plot. Any other plot will be hard to make, and box plots are ideal. This is the only way of reducing large number, millions of data points to something that you can read. Bar plots, very often used in biological research. I would say too often, because there are good alternatives to bar plots, and bar plots are not always adequate. I will show you examples where bar plots are really good. First of all, when you have a categorical variable like this, the variable is a strain of your cells or animals, wild type and free knockouts. And then we have 2 different categories, subcategories within each category. There are 2 different types of treatments, DMSO and some drug called DD342. You can see the proportion of cells counted in an instrument with arrow bars. This is a very correct and very appropriate bar plot. Another example is when you have a probability distribution, where your category is the count here, and you plot the probability. The important thing about bar plots is they should be used only with a quantity which is additive. Count is additive. If you look at the area of here, it will be proportional to the count, and counts from 2 different experiments can add together, and you can do it visually. It's even better here. The total area, this darker area here, would correspond to the total probability from these 3 bars. Another case of a bar plot is when you use a continuous variable, but then you divide all possible values of this continuous variable into bins. In this case, you plot a histogram of a p-value. And there are thousands or even tens or hundreds of thousands of p-values in this experiment, and you want to see how they are distributed. So a histogram here, you have count here or probability density, depending what you want to do. So again, you have additive quantities, and the area under one bar, area under the other bar, they represent count. If you add this area visually, it will represent the total count. The important part here is that you divide all possible values into distinct bins. There are no gaps between bars because this bin stretches from 0 to 0.1 and contains all the p-values between 0 and 0.1. This one extends from 0.1 to 0.2 and contains all p-values between 0.1 and 0.2. If you add some sort of gaps between these bars, it will mess up the plot and make it difficult to interpret. There are two ways you can plot the y-axis here is either count, which is a direct count, or you can plot density, which does not depend on the total count you have. It's count divided by total count times the bin width. The total area under the density plot is 1. This is the bin width. It's quite important to remember what sort of bin width you can select. If it is too small, the plot will look very messy. There will be lots of just individual bins going up and down. If you make it too large, you just lose information. The very, very important thing is that bar plots always have to start at 0, like in the left plot. On the y-axis, you have 0 extending to about 800, and there is a count in 2 different experiments. And visually, you can see that in treatment, the area of this bar is about twice the size of this bar. So visually, you see there are twice as many counts here. If you shift your axis in a way that 0 is down there and you reduce it to between, let's say, just below 400 and up to 800, you make a plot which is very misleading, because visually the control is very, very small by comparison to treatment, while in real life it's just factor 2. So this plot is really, really bad. Yeah, this is actually an example from, I don't recall where it came from, but it was from some, I think either a newspaper or a magazine, where there's a comparison of 1 million versus 17 million with 2 bars that mean absolutely nothing. Very misleading, very bad type of a plot. Don't do it. The second thing is you never should do bar plots in logarithmic scale, because there is no 0 in the logarithmic scale. Well, there is 0, but it corresponds to the count of 1, and it's nothing special. So if you plot it in logarithmic scale, you can choose the beginning of a plot anywhere, and depending what you choose, the plot will look differently. The area here is not proportional to the count. It's proportional to the logarithm of the count minus logarithm of 1. It's arbitrary and it's very misleading. Real 0, don't do this, ever. There are more problems with bar plots. This is an example of data showing the speed of growth of a microtubule in a cell in micrometers per minute. There are 6 samples here. If you plot it like this, well, first of all, the bar area, it's proportional to the speed, but this is not something which would be additive. If you look at 2 bars next to each other, the total area doesn't tell you anything. There is nothing like a total speed from 2 different experiments. You can't add it. Secondly, because bar plots have to start from 0, you can see that there is little variability here. You can perhaps amplify it like this. Ignore bar plots, just plot a point, plus minus arrow bar. Now you can compare these data points directly to each other. You can see that if this one stands out, others are a bit lower. You can see where arrow bars begin, where they end, which is rather hard to see in here. Or even better, if these data come from individual replicates, from individual experiments, it's better to plot actual data. And now you see, if you make this with an arrow bar, you will see that there is a big gap here, and the impression for the reader would be that this is completely above everything else. these data are very different. However, if you look at how data were collected, well, yes, it's up here, but there are some outliers down there which are at the same level as other data set. And even if it is statistically significantly different, you probably need to treat it with caution because there is a lot of overlap between sample one and sample two. Another bar plot, which in my opinion is problematic, it's just visually very messy. There is this repeated pattern of black and white bars. It's not easy on the eye. It's not easy to read. It masks actual data. Another problem is you can see only upper arrow bar, lower arrow bar is invisible, it's hidden. The bars are rather crowded. There is this confusing visual pattern. Again, it's not count based, it's not proportion based. This is distance, so bar plot is not the best. Also, this is a continuous variable time. These two bars belong to time 0. These two bars belong to time maybe 4 hours or so, and yet they are shifted. This suggests that white bars are shifted in time with respect to black bars. It's just because of the way it's presented. So I would say this is a bad plot. Don't do it. It's much easier to show it like in the lower panel, individual data points with arrow bars using different colors. And because they overlap here, maybe just for guidance, join them with gray lines. When you look at this plot, you immediately see what's going on, while the top plot lacks clarity. Well, and my favorite pet hate is something which is called dynamite plots. This is a plot you can see in literature. Unfortunately, even now, as we speak, there will be papers published showing plots like this. The main reason is because people use software packages where this sort of a plot is default. What happens here is the lower arrow bar is invisible. It's hidden. You are hiding something from the viewer. Why would you do this? I mean, what's the point? This is dynamite plot because they look like sticks of dynamite. And every time you make a dynamite plot, a puppy dies. Remember about this. You can improve this plot quite a lot. First of all, you can show full error bars. This is a bit better. But you can always also see that these data are normalized to wild type, and wild type is 100% here, which is completely redundant information. You don't need to show this. If it is divided by the wild type, just show your treatments T1 and T2. And again, bar plots are unnecessary. You can just show a very simple plot like this, two treatments, value with error bars. Or perhaps you don't need a plot at all. If you have only two values with error bars, you can report them directly in the text of your paper. This is an example of what I consider one of the worst bar plots ever. It comes from this website, the conversation, and there was an article whether double dipping of crisps in South is a food safety problem or it's just a nasty habit. They measured a number of bacteria. If you do this, how whether bacteria can spread from person to person and so on. And they showed data in this plot, and it's a beauty. It's so bad that it's beautiful. First of all, it's completely unnecessarily. It's a 3D plot, which doesn't help understanding data at all. Secondly, well, it's hard to say what categories are because this category is named, this one isn't. This is category which is named salsa bite dip, this is not. So there are some category descriptions missing. Then you notice one really curious thing. This bar, it extends to 1000, but on the axis it says 300. So something weird happened here. Even if you compare bars to the lines here, it doesn't make any sense because this is 158 and it extends to exactly 150. Only if you look very carefully, it's the projection, the sort of a wall with scale at the end is shifted back. So this, you have to extrapolate and find that it corresponds to the axis somewhere here, about 158. I mean, this is really, really bad, and I would vote it one of the worst plots ever. Everything here is wrong. Another section, pie charts. You might have seen pie charts. My answer to pie charts is very simple. No, just don't do them. Pie charts are awful. Make a bar plot, it's much better. In a pie chart, sometimes it's very difficult what the size of a pie is. It's just wrong. Don't do it. Here in this slide, I collected everything. This is your 10, 11 commandments of how to make good graphs. I'm not going to read them now. You can keep it later. You can find it in the handouts and come back to this if you need it. And similarly, recommendations specifically for bar plots. Bar plots are often used and they don't have to be used that often. And very often bar plots are abused in a way that makes plots very difficult to read. Okay, let's take a short break and talk about William Playfair. Have you ever heard of William Playfair? He was born in Lith, near Dundee, just to the west of Dundee, a little village. And he was a man of many careers. He was a millwright, engineer, draftsman, accountant, inventor, silversmith, merchant, secret agent, investment broker, economist, statistician, pamphleteer, translator, publicist, land speculator, blackmailer, swindler, convict in this particular order, banker, editor, and journalist. He invented line graphs, bar plots, and pie charts. He's quite famous for this. If you never heard of him, now you know. And this is I believe this is the first bar plot ever. It's very hard to read. I'm sorry, I shouldn't be really showing this, but this is the original bar plot. The second part of the talk is about quoting numbers and errors. Without plots, if you have to show a number, like this one, for example, well, you obviously don't want to do this. And there are certain rules of showing and quoting numbers. In a publication, you would typically quote the best estimate of your value plus minus some kind of error. And error can be, as usual, standard deviation, standard error, confidence interval, or some sort of a complex derived error from calculations. Make sure you tell the reader what type of errors you use every single time in a figure caption, or if you quote a number like this, then in brackets, let's say 95% confidence interval. The important thing to remember is about significant figures or significant digits. They are significant figures are those that carry meaningful information. Have a look at this example. A microtubule has grown 4.1 micrometers in 2.6 minutes. What is the speed of growth? If you divide 4.1 by 2.6 and use either a computer or a pocket calculator, if you still have one, you will get a number like this with lots and lots of digits. The problem is that most of these digits are just noise. It's completely meaningless. There are two significant figures in length and in speed. Therefore, you do not expect more than two significant figures in the result. There is no way of obtaining a more accurate result than your measurements at the end. So I would write this down as 1.6, just round it to the nearest to two significant figures, and write it down as 1.6 micrometers per minute. Remember, you can only quote significant digits. Everything else is noise. It's irrelevant. Now, how do you write down significant figures and make the reader aware of what is significant and what is not? So first of all, leading zeros are never significant. So these three numbers, 34, 0.34 at 0.000034, they carry the same amount of information. There is a 34 and there is an order of magnitude. You could write it down as 34 times 10 to a certain power. That's the whole information. Now, trailing zeros are a little bit more complicated. In this table, I'll show you a few examples of a number written down and how many significant figures it carries. If you write down 365, obviously there are 3 significant figures and they are considered significant. So this is quite an accurate number within 1/100th or so. If you write down 1.893, there are 4 significant figures. But now if you write 4,000, this is becoming a bit complicated. If you just write down 4,000 without any context, it could be either one significant figure, 4, and zeros are because you round it up, or it could be exactly 4,000. If you say that there are 4,000 people gathered in the market square, you probably don't mean that there were exactly 4,000, not 4,001, not 3,999. You give an approximate number and you can guess it from the context. However, if you say that we counted 4,000 cells in the device that counts cells, then maybe it's exactly 4,000. So depending on the context, that can carry anywhere between 1 and 4 significant figures. To make it absolutely sure what you mean, you can write it down as 4 times 10 to the power 3. And then there is only one significant figure, is marked here as bold. If you write it as 4.000 as time to the 3, this is a bit weird, but sometimes it might be necessary to convey that there are actually 4 significant figures, that all these zeros are meaningful, they are all significant. If you write it down like this, with trailing zeros here after the decimal dot, there will be 6 significant figures. But again, the leading zeros, as I said, they don't matter. So this number has only 2 significant figures. If you add 2 trailing zeros, then you notify the reader that there are 4 significant figures and they are 3, 4, 0, 0. They are all significant. There are rules of rounding numbers. It's quite simple, actually. You remove all the non-significant figures by rounding. So if you have a number 1,234 and only 1 and 2 are significant, you can quote it as 1,200. This one will be 1,300 because you round it up. Figures from 0 to 4 are round down, from 5 to 9 they round up. So this 3 is rounded down, this 8 is rounded up, and similarly here. Now, how do you know how many figures are significant? Well, to find how many significant figures are in the number, you need to look at its error. So you use sampling distribution of a standard error, and if you do this, we can find that error of the error is like this. This is just the only formula I'm going to show you today. And you can then estimate how many significant figures are, okay? Have a look at this example. Let's say you have a sample of 12. The standard error, if you calculate from this sample, is this, but obviously some of these figures are not significant. So what you can do is then calculate a standard error of the error according to this formula. And if you do this, the error of the error is about 4.9 or about 5. Which means that your standard error could be written like this. This is still not a correct description, but the standard error is about 23 plus minus 5, which means that only these 2 or maybe even 1 figure is significant. You can only trust 2 because the next figure, 3, has an error of about plus minus 5. So you should round the standard error to about 20. Instead of saying 23.17345, you would say 20. You don't have to remember this formula and all these calculations. You can just remember this. This is a very simple table showing the sample size. This is the relative standard error and how many significant figures you need to quote, which means that if your sample size is about 10 or less, you can only quote 1 significant figure of the error. Okay. If your sample is 100 or more, then maybe 2. If your sample is very large, you can quote 3 figures, which means if you quote your number with 3 significant figures like this, This is error. We are still talking about errors. There are three significant figures, 1, 6, and 5. If you write down this, then you implicitly state that you have 10,000 replicates, and you probably don't. You probably have a 10 or so. So this should be shortened and rounded. And how do you do this? Well, like this. First, you need to get a number and its error. Then you have to find how many significant figures you have in the error. You start with the error, and in most cases, it will be only 1 or 2. 3 would be very rare when you have lots of samples, typically 1 or 2. And then you align the error and the number at the decimal place, like in this case. This is your number, 1.23 and something else. And this is error, 0.02 and then something else. But we quote only one significant figure of the error, and we cut off the number at the same decimal place. And then we quote 1.23 plus minus 0.02. If you don't have a decimal place, you just align them together and keep, let's say, in this particular case, 2 significant figures of the error and 2 significant figures of the number. And then you would quote it like this, 75,000 plus minus 12,000. Definitely not like this. The examples on the right are all incorrect. The correct version would be this. Incorrect would be quoting shorter error number and longer error. They are incompatible with each other. Or the other way around, like this. This is to make sure that you know which figures are significant. If we have 2 of them, we have to write down 6.0, not 6. I discussed this a moment ago. And in some other cases, when your numbers are either very large or very small, you can use times 10 to the minus 5, and you put errors in brackets. What would you do if you had a number without error? Suppose you have a number without error, and I would say go back to your lab and do more experiments, but it's not always possible. So it depends on the context again. If you make a statement in your paper that centromeres are transported by microtubules at an average speed of 1.5 micrometers per minute, you quote 2 significant figures and you say, okay, these are 2 significant. So this is probably, this 5 is uncertain, maybe it's 1.6, maybe 1.4. We don't quote a formal error, but we say to the reader what accuracy of this result is. You can say the new calibration method reduces error rates by about 5%. So again, the number we quote here implicitly tells you what the error is, and it's usually at the last figure. And all quoted figures like 30 minutes, 22 degrees, all quoted figures are presumed significant if you quote a number without error. Avoid computer notation. If your computer gives you a result that p-value is 5.51 e minus 1/4, don't do it. Round it up and say 6 times 10 to the power minus 14. This is a bad example. This is again from some software that created this sort of axis, which is very hard to read. It's much better to use notation like this: intensity times 10 to the 6, and it's 0, 10 times 10 to the 6, 20 times 10 to the 6, and so on. It's much easier to read. Fixed decimal places are bad. If you copy something from Excel, your raw data might be rounded like this to 1 decimal place. The problem is that you are beginning to lose information. You quote too many significant figures, and at the end there are no significant figures at all. You have only 0, while actual number is 0.02. So you should quote it like this, assuming there are 2 significant figures in each result. This will be 1.5 times 10 to the power 4. This would be 2200, and so on. Finally, a summary: how to quote numbers and errors. When you know error, just again, first calculate the error and its uncertainty. This will tell you how many significant figures of the error to quote. Typically would be 1 or 2. Hardly ever you are getting 3 or more significant figures. And then quote the number with the same precision as the error. So you start with the error. And there are a few examples here. When you don't know error, well, you still need to guesstimate how accurate your data are. Quote only figures that are significant. If you find a p-value in a statistical test, in most cases, you have only one or maybe two significant figures. If you get from your computer a p-value like this, do not quote it. Just say 0.03. Use common sense. Try estimating order of magnitude of your uncertainty. For example, if you measure distance between two spots in a microscope, and the software will give you this number, 416.23 nanometers, but you know that the resolution of your microscope is only 100 nanometers, then obviously you should just quote 400, not this number. Thank you.